Hello everyone. Coming to the latest developments in this microbiology and biochemistry field, I'm just concising it to 5, but we have many more in this field. Coming to those 5 recent advancements, CRISPR-based microbial genome editing for targeted drug production, development of synthetic microbiomes for disease management, AI-driven microbial identification and classification, enzyme engineering for sustainable biocatalysis process, real-time metabolic tracking by biosensors. Let us discuss about the first one, that is CRISPR technology. CRISPR is nothing but a clustered regularly interspaced short palindromic repeats. It's a gene editing process where we use microorganisms. The wanted gene is inserted into the microorganism gene so that it can produce multiple genes with the required genome. Okay, so using CRISPR, scientists precisely edited microbial DNA. We can insert the genome, or we can delete, or you can modify the gene. And for this, we require a RNA, guide RNA, Cas9 enzyme, and DNA, which can cut the DNA at different locations. So nowadays this engineered microbes are used in the production of insulin, anti-cancer drugs, as well as antibiotics and also enzyme synthesis. CRISPR is a faster and more accurate process when you compare with the other processes. So as we discussed in the previous slide, we have Cas9 enzyme, a guide RNA, and a matching DNA. This Cas9 enzyme is used for the unwinding of a section of DNA, and after unwinding, this is what we call, as you know, it's a double-stranded DNA which is twisted. So to unwind this double-stranded DNA, we require Cas9 enzyme. After this unwinding, this guided RNA will bind with the matching DNA at specific site where its genes will match with the, or its codons will match with the RNA codons. This fragmented DNA or repaired DNA is, or edited DNA is inserted into the microorganism, which is multiplied in the microorganism and produce a lot of genomes. Coming to the CRISPR technology, let us discuss 2 examples. Insulin was extracted from animal pancreas. As you know, it is extracted from pigs and cows' pancreas. But we can use this CRISPR technology to produce more insulin. And in this CRISPR technology, we insert human insulin gene into the DNA of E. coli. And this modified bacteria is multiplied, and it is grown in the large bioreactors so that it can produce multiple DNA with this modified gene. Next, we are using Streptomyces. It's a natural producer of antibiotics. Especially, we use this Streptomyces bacteria in the synthesis of erythromycin, which is an antibiotic. And using this CRISPR technology, we can also multiply, or you can boost the erythromycin production. You can create a new antibiotic derivatives by using this. And you can also overcome antibiotic resistance, which is a common problem nowadays. Next, synthetic microbiomes for disease management. Actually, there are 3 categories. One is microorganism, microbiota, and microbiome. So microorganisms, as you know, these are tiny organisms which are living in all kind of environments. Microbiota is a community of microorganisms which live in a specific environment. Microbiome, it's a community of microorganisms, and they play specific role in the environment wherever they are located. Okay, so synthetic microbiomes are lab designed microorganisms which are tailored to produce a specific enzyme or to function as a specific activity. Generally in diseases like inflammatory bowel syndrome or inflammatory bowel disease, obesity, even in mental health disorders, all of these are linked to dysbiosis. This is nothing but imbalance in the microbiota. You might have heard more about this gut microbiota and all. This gut microbiota and different microbiota which are present in the human body play a major role in controlling of the many diseases. Scientists have identified beneficial microorganisms. They have modified their functions by using CRISPR technology and some other technologies as well. And they have designed a stable microbial consortia. This synthetic microbiota can survive in various temperatures and various environmental conditions, and they can outcompete the harmful bacteria. They can produce therapeutic metabolites as well. They can regulate the immune responses. Coming to the examples, engineered microbes are tested for mouthwashes to suppress the cavity causing bacteria like Streptococcus mutans, which may prevent the caries, which is replacing the traditional antiseptics. A synthetic E. coli Nissel strain is engineered to break down phenylalanine. This phenylalanine is a amino acid which causes phenylketonuria. This PKU, this engineered E. coli strain can prevent the brain damage in PKU patients without needing for ultra strict diet.
Vishnu Audio Visual Center, like and share and subscribe and click bell icon.